Hi, my name is Maya Munstermann. I'm a fourth-year PhD student in the Okamoto lab, and my research focuses on how climate change is affecting kelp forest ecosystems. In around 2014 to around 2016, a series of marine heat waves hit the California coastline, and this elevated temperatures far beyond what was normally seen on the North Coast. When this marine heat wave came through, it was kind of like a perfect storm of things going wrong. The warm water weakened the kelp, which is the foundational species of the kelp forest ecosystem. This warm water was also associated with a disease that came through and killed all of the major sea star predators in the system. And what they do is that they eat the urchins in the system. When the marine heat wave came through and the disease wiped out all of these major predators, now there was nothing controlling the urchin populations. And so urchin populations exploded, and what remaining kelp that there was then just kind of mowed down by these urchins that were out of control. What my research is focusing on is the transition between kelp-dominated states to urchin barrens. It kind of looks like a carpet of purple spines on the ocean floor, where all of the diversity and structure that you would normally see in a kelp forest is now pretty much this barren landscape. What a typical day looks like for me in a field setting: we wake up very early to catch those low tides. We gear up, we put on our mask, we put on our really thick wetsuit, and then we go out in the water and do our collections. And so we have these slates with waterproof paper and our pencils, and we swim out and we collect measurements on all of our kelp individuals that we're interested in looking at. We're also collecting urchins to see what is going on with the urchins at the same time. Being connected to the ecosystem that you work in is, in my opinion, crucial to understanding what's going on. If you can be in the field, you can notice these slight changes that happen over time. After we collect all of our measurements and our urchins, we drive back to BML, which is the Bodega Marine Lab in Sonoma County. So we dissect all the urchins and preserve the samples for future processing. When we look at the urchin, we take histology, so understanding what's happening at the cellular level of these organisms. And then the other type of sample we take is genetic samples. So that leads me into what a day at Berkeley is kind of like, where I'm doing a lot of genetic work now, trying to understand what the gene expression is of these animals under climate change. What Berkeley offers is the resources and the lab space to do my genetic studies. I feel like no one really talks about this, is that this kind of work is really expensive. And without the generous support from donations and donors, I wouldn't be able to do this work. Without being able to purchase the gear or the transportation or sample processing, none of this could happen without funding support. I hope that this work will provide stepping stones for prioritization. Targeted urchin removals can happen in places where they will have the most likelihood of controlling the urchin population. There's no way to protect every single stretch of coastline. What my kelp growth work hopes to do is look at what sites of kelp has the highest capacity for regrowth, and then focusing conservation efforts on those sites. No matter what I end up doing in my life, the driving theme for me is trying to understand how humans are affecting marine ecosystems. My long-term goals would hopefully be a professor where I can continue doing research, I can mentor incoming students, and work with stakeholders in the matter as well. So community members, tribal members, and governments working together and trying to understand how can we all coexist in this space, and how can we make sure that there is not drastic changes that are going to change the ecosystem irreversibly.